Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 16th February 2021. As all of us know that we are into the revision mode and we are revising in the form of sample papers, CBSE sample papers. Already we have solved 3 sample papers. This is the 4th one. In the 4th one, in our previous session, yesterday's session, we answered up to section C. Now, in this session, we are going to answer, discuss, reflect upon section D. As all of you know, section D is long answer type questions. And this consists of 3 questions and each question carries 5 marks. So totally, section D will have 15 marks weightage. And in this, what is important is you need to be specific. Since it is long answer type, you are tempted to write lengthy answers. So no stories. Always 2 things you must remember whenever you are answering long answer type questions. That too something like say 15 to 20 sentences. Number 1 that you need to focus is write point wise first. And below that point, you keep on expanding the points that you have written above. And substantiate your answer with a beautiful diagram. He said that spermatogenesis is the genesis means birth of a sperm. Spermatogenesis, it occurs in males starting from puberty till the completion of life cycle, whereas oogenesis, it starts even before birth during embryonic development and occurs till the lady reaches menopause, somewhere say 35 to 40 years. And spermatogenesis, a single spermatogonium after second meiotic division forms 4 haploid spermatids, haploid, one set of chromosomes N, and that mature to form spermatozoa, spermiation we said. And then a single oogonium after second meiotic, see meiotic 1, meiotic 2, produces only 1 functional ovum and 2 non-functional polar bodies which will be disappearing. Third point, the process of spermatogenesis, that is second meiotic division, completes in testes and releases mature sperms, release of sperms, spermiation. The second meiotic division of oogenesis completes in fallopian tube when sperm enters the secondary oocyte. In that question 2, draw a sectional view of human ovary and label the different follicular stages, ovum and corpus luteum. I said yesterday that this diagram is very important. This is there in practicals also. This is worth 3 marks. You need to draw and label. And do not forget to draw beautiful diagram. And then label all the follicles, primary follicle, secondary follicle, tertiary follicle, Graafian follicle. And then release of ovum, ovulation. And once the ovum is out, ovulation is done, what is left out is corpus luteum. And corpus luteum, you know, is very important. It secretes progesterone. Keeping that in our mind, suppose you felt difficulty in answering that, then you have a choice. What is the choice question? The events of a menstrual cycle are represented below. Answer the questions following the diagram. That he has given you luteal phase 16th to 28th day, of course menstruation 1 to 5th day, after menstruation of 1 to 5th day, then follicular phase 6th to 15th day, then follicular stage is followed by luteal phase 16th to 28th day. Based on the diagram, there are questions. Question 1. State the levels of FSH, follicular stimulating hormone, LH, and progesterone simply by mentioning high or low around 13th, 14th day and 21st to 23rd day. Of course, here you can understand that it is 13th to 14th day comes under follicular phase. So naturally follicular FSH has to be high, LH also has to be high. And 21 to 23rd day it enters into luteal phase, and during the luteal phase it has to be both FSH and LH have to be low. And then question progesterone, of course it is again low, and 21 to 23rd it has to be high. Question 2. In which of the above mentioned phases does the egg travel to fallopian tube? Of course all of us know it is during luteal phase. And third, why is there no menstruation upon fertilization? There is no menstruation upon fertilization because the ovum which was to shed gets implanted. Once implantation takes place, no more menstruation. Uterine wall and blood vessels maintain the embryo. There will be a lot of changes in the uterine wall. Question 32. The development of bioreactors is required to produce large quantities of products. Question 1. Give optimum growth conditions used in bioreactors. Of course, optimum conditions: temperature, pH, substrates, salts, vitamins and oxygen. And second one, draw well labeled diagram of simple stirred tank bioreactors. So that is the reason why I said you must practice diagrams. And after drawing the stirred, there are two types of bioreactors. He is asking you stirred tank bioreactors. So here is the diagram. So on top there is a motor, there is a foam breaker, flat blended impeller will be there, and there is a culture broth and the sterile air pumped in, acid base for pH control, there is one inlet or outlet, then steam for sterilization outlet also is there. So all these are to be drawn and labeled. Question 3. How does a simple stirred tank bioreactor differ from sparged stirred tank bioreactor? The stirrer facilitates even mixing and oxygen availability throughout simple stirred tank bioreactor. Whereas in case of sparged stirred tank bioreactor, air is bubbled throughout the reactor for proper mixing. Now you got a choice question. Explain how to find whether an E. coli bacterium has transformed or not when a rDNA bearing ampicillin resistant gene is transferred into it. When E. coli bacterium is transformed with a rDNA, recombinant DNA bearing ampicillin resistant gene in its plasmid, the recombinant plasmid will lose tetracycline resistance due to the insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on ampicillin containing medium. The transformants growing on ampicillin containing medium are then transferred to a medium containing tetracycline. The recombinants will grow on ampicillin coating medium but not one that containing tetracycline. This is important, not containing tetracycline. But non-recombinants will grow on the medium containing both the antibiotics. This is how we can convert it. Question 2. What does the ampicillin resistant gene act as in the above case? In this case, one antibiotic gene helps in selecting the transformants, whereas the other antibiotic resistant gene gets inactivated due to insertion of alien DNA and helps in the selection of recombinants. Ampicillin resistant gene in the above case helps in selecting the transformants and act as selectable marker. We come to the last question. Explain the interrelationship between organic farming and biofertilizers with the help of any 3 suitable examples. Farmers using organic farming use biofertilizers, organisms that enrich the nutrient quality of the soil, instead of the normal chemical fertilizers that normally farmers apply. The main sources of biofertilizers are bacteria, fungi and blue-green algae, cyanobacteria. Example, rhizobium in the root nodules of leguminous plants symbiotic relationship, fix atmospheric nitrogen and enrich the soil. Other free-living bacteria which can fix atmospheric nitrogen are Azotobacter, Azospirillum. Mycorrhiza, which is a symbiotic association between fungus of genus Glomus and roots of higher plants, absorb phosphorus from the soil. They also make the plant resistant to root-borne pathogens, tolerant to salinity and drought, and an overall increase in plant growth and development. Cyanobacteria, autotrophic microbes, fix atmospheric nitrogen and increase organic matter in the soil. Anabina, nostoc and oscillatoria. This is a beautiful one, not only for exams, even for our daily life. Particularly children of farmers, please do tell your parents to go for this. Last, choice question. How is primary effluent treated in a sewage treatment plant before it can safely be released into the rivers or streams? Again, this is also not only for exam sake, even in our day-to-day life, it is very, very important. Sewage, through filtration and sedimentation, forms supernatant part in primary effluent, and all the solids that settle down from the primary sludge. Secondary treatment or biological treatment of primary effluent involves different steps. Number 1. Primary effluent is passed into large aeration tanks. Second, it is constantly agitated mechanically and the air is pumped into it. Third, this allows vigorous growth of useful aerobic associated with fungal filaments to form mesh-like structures. Fourth, the microbes decompose the major part of the organic matter in the effluent. Fifth, it reduces BOD, biological oxygen demand, of the effluent. Sixth, BOD refers to amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. When the BOD of sewage is reduced significantly, the effluent is passed into a settling tank where the bacterial flux are allowed to sediment, forming the activated sludge. A small part of activated sludge is pumped back into the aeration tank to serve as the inoculum. Inoculum is just like your mummy uses a small amount of curds. She adds into the milk so as to convert the milk into curds next day. That small dose of curd she is adding, that's inoculum. The remaining major part of activated sludge is pumped into large tanks called anaerobic sludge digesters. Last, here, another type of anaerobic bacteria digest the bacteria and fungi in the sludge, producing methane, CH4, hydrogen sulfide and carbon dioxide, which forms biogas, major components of biogas. And this biogas can be used as a source of energy as it is inflammable. So every product that is formed is very, very useful. Hence, it is always good to use the STP,